Hi everyone, this is Dr. Masab Umair and I welcome you to our YouTube channel Viral Insights. Today I'm going to discuss our article which is on the serotype and genotype diversity of dengue virus during the 2023 outbreak in Pakistan, and this article was published in the Journal of Medical Virology earlier this year. Dengue it stands as a major public health problem in tropical and subtropical countries of the world and affecting nearly half of the world population every year. And it is estimated that it causes around 36,000 deaths annually. So the virus is a flavivirus and belongs to the family Flaviviridae. And there are four serotypes of the virus, which have around 65 to 70% of homology. So infection with one serotype, it provides lifelong protection against that particular serotype. However, it does not provide protection against the other serotypes. So looking at the dengue virus spectrum, disease spectrum, so majority of the cases they are asymptomatic and cause mild dengue fever. However, a small percentage of infections can lead to severe disease such as dengue hemorrhagic fever and dengue shock syndrome. So there are different risk factors that lead to secondary infection, but the most important is infection with the dengue serotype that is different from the primary infection. In 2023, a total of 643 million cases and 6,892 deaths were recorded due to dengue globally, with around 56,000 cases and 28 deaths per million population. If we look at continent-wise, the highest number of cases were reported from South America, with the highest number of deaths in Asia. The highest number of cases and deaths per million was reported in North America. If we look at the disease situation in Pakistan during 2023, so dengue caused a high number of infections and cases last year, and around more than 20,000 cases were reported in Pakistan. So, looking at the high disease burden and outbreak of dengue last year, so we investigated the dengue outbreak by generating whole genome sequence data to assess and explore genomic diversity and track virus evolution over time. The methodology of our study, so during July to October 2023, a total of 153 blood samples of NS1 confirmed dengue patients that were tested in different hospitals of Islamabad and Rawalpindi. They were referred to our Department of Virology at NIH, and total nucleic acid was extracted using MagMax viral and pathogen nucleic acid isolation kit and the Kingfisher Flex purification system. The serotyping was performed using the CDC multiplex serotype specific real-time PCR protocol, and the nucleic acid, viral nucleic acid, was treated with RNA-free DNAs and prepared for unbiased pair and sequencing using the NAB Next Ultra 2 directional RNA library propagation kit. For NGS analysis, quality of raw NGS reads was assessed using the FastQC, and low quality sequences and adapters they were removed with the Trimomatic. The Picard tool was used to remove PCR duplicates from the filtered reads, which were then assembled into contigs with the SPIRS program and compared to the NCBI database to map the reads with the most related genome using the BWLN. For variant calling, BCF tools call was used and consensus sequences were generated with Genus Prime. And dengue genotypes, they were determined using the flavivirus genotyping tool that is available online. For phylogenetic analysis, the high quality whole genome sequences were selected. Blast search was conducted on NCBI database against each of the current study sequences, and study sequences containing more than 2% ends or near identical matches were excluded from analysis. Similarly, multiple sequence alignment was performed using MAFT, and we used Mega 11 for phylogenetic tree analysis, and pairwise distances for heat map were calculated using the same software. So between July and October 2023, the samples received, which were around 153 samples, basically they were NS1 positive, and they were received from different hospitals of Islamabad and Rawalpindi, as I mentioned previously. So we performed descriptive analysis, and it showed that the mean age was around 32 years, with more than 95% of patients that aged between 11 and 60 years. The male to female ratio was 2.6 to 1 ratio. Among the patients, 63% were residents of Rawalpindi, while 37% were from Islamabad. Clinical and hematological data was available for 73 patients with fever and myalgia reported in all the patients, and backache was present in only 4% of the cases. All NS1 confirmed dengue samples, they underwent serotyping, with 64% successfully serotyped using the multiplex real-time PCR. Of these, 85%, which is equivalent to 83 cases, were identified as DEN1, whereas 15% were identified as DEN2 serotype. The month-wise distribution of these samples showed a predominance of cases in September and October. Further analysis of weekly distribution highlighted an increased number of DEN1 and DEN2 cases during the second and third weeks of September 2023. Our analysis, it revealed a substantial shift in serotype distribution, with 85% of cases that were attributed to DEN1 and 15% to DEN2. And this is, in contrast to earlier studies from Pakistan, where DEN2 was more predominant, especially in 2022, and previously as well as in the years 2019, 20, and 21. In the current study, we successfully obtained whole genome sequences of 11 of the 23 samples. Within this subset, 17 sequences were from DEN1, and one whole genome that we retrieved was of DEN2. So if we can see that among these 18, a majority of these cases, they were from Rawalpindi, but we also managed to sequence DEN1 virus from Islamabad. And if we see, the DEN1 serotype was classified as genotype 3, whereas the DEN2 serotype had the cosmopolitan genotype. So this is on the left side is a maximum likelihood phylogenetic tree of DEN1 whole genome sequence, and the study strains, they are highlighted in red triangle. On the right side, the figure, it shows pairwise distance represented by heat map. So our results showed that as per the flavivirus genotyping tool, all of the 17 whole genome sequences of DEN1, they belong to the genotype 3, and the DEN1 viral sequences from the current study, they clustered with viruses from the 2022 outbreak in Pakistan, exhibiting high nucleotide sequence identity, which was around 98.8% to 99.7%. So, and these study sequences also showed high nucleotide similarity with strains from US, which were detected in 2022, Singapore and China detected in 2016, and from Bangladesh detected in 2017. The DEN1 virus of 2023 outbreak, which is our study sequences, they cluster with strains from the 2022 outbreak in Pakistan, sharing high nucleotide similarity. And this was the same from Islamabad and Rawalpindi region. So this indicates the persistence and circulation of genotype 3 in the region, and because this genotype was also previously detected in 2019. So we had just one DEN2 whole genome. So the study sequence is highlighted here in red triangle. So our results, they showed that the DEN2 sequence belonged to the cosmopolitan genotype, and we identified the clade as the 4A1 clade. Genetically, the cosmopolitan Den2 sequences from the current study, they shared a distant identity with cosmopolitan genotype from Pakistan that was previously reported during 2008 to 2013. Additionally, Pakistani Den1 virus, it showed higher nucleotide identity with viruses that was circulating during 2018 and to 2019 in China and Singapore. Looking at the mutation analysis, the DEN1 study viruses exhibited high sequence similarity of 99 to 100% at amino acid level. In comparison to the reference sequence, a total of 34 amino acid substitutions were identified in Pakistani DEN1 sequences with 9 mutations in the structural proteins, capsid, membrane, and envelope. However, within the coding region of study sequences, no nucleotide insertions or deletions were detected. And if we look, mutation analysis of our study DEN1 sequences in comparison to our previous sequences from Pakistan collected during 2019 and 2022. So we saw 6 unique mutations. In comparing the DEN2 sequences with the reference strain, a high number of amino acid changes were observed. The study DEN2 sequence, it differed significantly from those in previous outbreaks from Pakistan that were reported during 2008 to 2020, 2013. Specifically, 2 unique mutations were identified. In case of DEN2 strain, both the E and the NS5 gene harbored common distinct non-synonymous mutations in both the 2022 and 23 outbreaks. And these genes, they play a key role in infectivity, antigenicity, genome replication, host protein interactions, and drug targets. So, in conclusion, our study basically shed lights on the evolving landscape of dengue virus in Pakistan, and it particularly, highlights the shift in serotype distribution that we saw, because in 2023 we saw the dominant predominance of DEN1, whereas comparing it to our 2022 outbreak, DEN2 was predominant, and as I previously discussed that the shift of the strain, it has implications on disease severity. So because infection with one serotype, it provides a lifelong immunity against that serotype, but it does not provide protection against other serotypes. So again, it's very important to do serotyping during different outbreaks, and to make sure that there is continued serotyping and genomic surveillance of dengue virus in Pakistan. Similarly, in our study, we also identified distinct non-synonymous mutations and the dominance of previously reported significant mutations in the currently circulating strains, which again warrants further investigations to understand the impact of these mutations on disease severity as well as the transmission of the virus. Thank you.